Before we get started here, I'd like you all to take a quick survey on the question, if you could pick the traits of your future child, would you? And the URL for the Survey Monkey will be here. I'm assuming it'll take like a minute for them all to take that. Okay. On August 6, 1945, the first atomic bomb was dropped on Hiroshima, Japan, causing thousands of deaths and devastating destruction. Once the knowledge on how to create such a weapon was released, it could never be taken back. And now the reality is, 9 nations today continue to harbor nuclear weapons as part of their self-defense. This acquisition of knowledge happened to release a Pandora's box filled with massacre and endless paranoia. Recently, scientists have found a tool that could change humanity's future. But for the worse or for the better? One thing's for sure, once this technology is used to edit our genes, it will change the world forever. Let's just hope the result of nuclear weapons doesn't foreshadow the possibilities of genetic engineering in human beings. Hello, my name is Elaine, and I am here to talk about about how not all progression should be embraced wholeheartedly without thinking about consequences. Genetics are the puzzle pieces that make up each and every one of us in this room. Who are we to rearrange the final product? To reach a form of compromise in which some of the benefits of gene editing can be enjoyed whilst most risks are avoided, I believe only certain areas of genetic engineering in human beings should be explored and tested until they can be safely applied using modern-day science, while other areas should be banned entirely to ensure humanity doesn't become too wrapped up in controlling their own evolution. So, to do this, first we're going to have a brief science lesson on genetic engineering and its different categories that are specific to this project. It's recent advances and current events, what you need to know, what's important to know, the hype, why do we want this technology so bad, what can it offer us, and then the morality, what's stopping us from employing this technology right now, and possible alternatives to using genetic engineering. So first, before we delve into a moral debate, let me explain exactly what genetic engineering is and the different categories you need to know. Genetic engineering can be defined as the editing of an organism's genes in order to manipulate a certain trait. We see the results of genetic engineering every time we step into the produce section of our local grocery store. So when you see those bright, ripe red tomatoes on the stand over there, you can pretty much assume that they did not naturally grow that way. They're so vibrantly red and so large because a scientist specifically picked to emphasize those traits to make the product appealing to the consumer. So they edited those traits to make sure that they were very prominent. Okay, so the two different categories are somatic cell gene therapy and germline cell gene therapy. So first, and that basically just means that there are two different types of cells you can take DNA from to edit. So first, somatic cells are any cells in your body that are not reproductive cells. And editing them means that the changes made only affect the individual the changes are made on. And the one problem with this is that the changes are kind of short-lived because your cells die and are replaced with new cells. But if you continue to get the procedure done, you'll continue to have therapeutic effects. The next is germline cell gene therapy, and germline cells are reproductive cells, so eggs and sperm. And the changes that are made are passed down to all of your descendants. And when the changes are made, they're made in the early stages of an embryo, and all of the cells of the developing embryo are affected. So, when learning about recent issues in genetics, specifically germline gene therapy, A phrase you will commonly encounter is genetic enhancement. It should be noted that genetic engineering and genetic enhancement are two different things. Genetic enhancement is taking the methods of genetic engineering and using them for things other than medical cures. It is the idea of improving traits such as height, strength, intelligence, and etc. Oftentimes, genetic engineering will be referred to as a slippery slope because people will think that it's going to start off with good intentions for developing cures for genetic diseases, and then it will kind of turn into an excessive use of unnecessary improvement, like changing and picking the traits of your future babies. So, why are we talking? I mean, to me, editing human genes sound like some fictional dystopian future that is thousands of years ahead of us. So why are we talking about it now? And we're specifically talking about it now because earlier in 2015, a technology was found or discovered called CRISPR-Cas9, which enables scientists to cut and paste sections of DNA more effectively and more accurately than ever before. This means that the future I thought was thousands of miles away could be in reality right around the corner. Here's a video to explain what CRISPR does from its very own founder, Jennifer Doudna.
A few years ago, with my colleague Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to diffuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells.
Alright, we're going to stop there. But one thing I really wanted to highlight is, I'm not sure if you caught it, on April 18th of last year, a study was published from a group of Chinese scientists stating their findings after testing on human embryos with CRISPR-Cas9. And although the embryos were non-viable, meaning they couldn't eventually become babies, a debate was still sparked by the public. Some thought an ethical boundary had already been crossed, but we will get into that more later. What they really found is that CRISPR isn't ready for the advanced measures they would like to take, which leads me into my next point. Sorry. I didn't mean to alarm or excite you, and I'm sorry if I did, but the reality is CRISPR really isn't ready for use in humans at this moment. When tested in complex organisms, it tends to have unpredictable results. In this specific experiment, the scientists attempted to use CRISPR to remove a gene that causes a fatal blood disorder, and it was performed on 86 embryos. Out of that, only 28 were correctly spliced, meaning that only 28 cut out the gene correctly and removed it. And even fewer, maybe just a handful of those, were able to insert the new gene, the new healthy gene. Another reason why it really just isn't ready for use in humans is it's only been a decade since the human genome was read. So Eric Lander, a geneticist and a professor of biology at MIT, wisely states in his article called The Brave New Genome that it has only been about a decade since we first read the human genome, what I have said there. We should exercise great caution before we begin to rewrite it. The scientific field focused on DNA and genetic engineering is incredibly fast-moving. Look how far we've come in genetics research just in a decade. But this also alludes to the fact that we still have much to learn about DNA itself. It is hard to correctly edit and manipulate something you don't fully understand. And this last part here is just that a hashtag was even made to try and tone down some of the hype called hashtag CRISPR and it was just people making jokes about CRISPR, and it's quite funny actually. So, why is it even worth pursuing? And why are we so excited about the prospects of germline gene therapy specifically? And really, there are some great things about it. The main reason scientists are even examining gene therapy is the hope that it can cure genetic diseases and help us gain a better understanding of more common diseases. Genetic diseases like sickle cell anemia occur because of a defect in one gene. If CRISPR-Cas9 could be used to alter this gene, we could have a cure for disorders like this one, and Huntington's disease, and cystic fibrosis. Editing germline cells of the people afflicted could remove the disease from humanity's entire gene pool, and we'd never have to deal with it again. It would just be gone, which is great. Another exciting hope for the use of CRISPR in humans is that it will help us make connections between certain genetics genes and diseases such as Alzheimer's and cancer. Before CRISPR, this process was a slow form of trial and error and testing in animals. Now the process can be sped up and connections found much faster. However, these harsh diseases may contain roots in hundreds of different genes, making it more difficult to foresee whether gene editing could offer an eventual cure. And the last one up here is kind of a genetic enhancement from a genetic enhancement point of view, but it's still interesting nonetheless and kind of cool. So the idea is that CRISPR could create a human race that could think quicker, run faster, climb higher, and feel deeper than ever before, and could lead humanity to stretch beyond its physical and cultural limitations that are biologically in place now, to stretch and become almost improved in every sense of the word. So by using genetic enhancement, we are reaching humanity's full potential and alleviating unnecessary human suffering, according to Nick Bostrom, a professor in philosophy and a director of the Future of Humanity Institute at Oxford. So what's stopping us from employing this technology now? And the answer is really morality. Some people are concerned with the morality of this entire germline gene editing process. And the first objection is that to take the step of engineering our own genes is to take human evolution into our own hands. It is essentially playing the role of God. Our species would be making monumental decisions on which genes in future generations would survive and which would die out. Genetics would never be up to chance again. This can go one of two ways. It can make a race of superhumans, or create a dystopian future where genetics is used as a weapon in order to form super soldiers. Another moral objection is the loss of individuality. So again, this is towards germline gene editing specifically. If selecting children's genes ever becomes a reality in the future, not only genes for physical traits would be determined, but also those for personality traits. A large part of the human experience is finding and defining oneself as an individual. If a parent picked out its child's personality traits, its journey of self-discovery no longer holds the same meaning. So basically, a parent is picking out certain traits and kind of defining who this child is before they're even born. And the question is, do we want that? Another objection is loss of freedom. So the children subjected to genetic engineering, they feel as though they have lost their freedom to create their own identity as an individual. The child's freedom of choice would be detrimented. Genetic enhancement decisions made by parents would end up steering their child toward certain life choices. And lastly, there would be a loss of equality. So genetic engineering would introduce a whole new way to discriminate. It would be pitted as the genetically engineered against the natural human beings. And since if this was ever employed, the procedure would be most likely expensive. That line would probably be drawn between rich and poor. But there is one moral support that I specifically found on genetic engineering, and that is that it's from a man named Julian Savulescu. He's an Oxford professor of philosophy, and he basically says that if we have the technology and knew that it was safe, it is our moral obligation to make our children the best that they can be. So, I mean, those are the moral objections, each of the moral arguments, and I'll let you decide who you side with. So lastly, we're going to talk about alternatives that you can use because a lot of the people who want genetic germline gene therapy are people who are want to have a healthy child and maybe contain recessive genes or dominant ones of harmful genetic diseases. So if that is the case and you would like to have a biological child, I've found two alternative options that you could use, and one of them is PGD screening. And that's basically they take different samples of your eggs and your sperm, or your spouse's sperm or your spouse's eggs, and they fertilize them outside of your body, and they test each embryo for the one that is least likely to be affected by these genes. Another is third-party reproduction, and that's having a sperm donor, an egg donor, and picking the traits from them so that you can kind of assure that you won't get harmful traits for your baby. And the last one is not for a biological child, but is still a viable option, and that's adoption. So the way that I've decided to apply the knowledge that I have acquired throughout this year is I've decided to write an editorial to advocate for my opinion to ban germline engineering, gene editing, and I have submitted it to the school newspaper, The Highlander, which will be released at the beginning of May. I am also currently attempting to submit it to our local Birmingham newspaper, so if you're all subscribed to The Eccentric, please keep an eye out. I'd really appreciate it. Remember, editing germline cells means the changes are passed down to every descendant. It's in your entire progeny forever. This means no mistakes can be made. Edits stick for forever. Although the rewards of gene editing are tempting, the risks that come out because of germline editing puts a damper on the excitement. This doesn't mean I think the field should not be explored and tested more. I fully support somatic cell gene editing, but I just don't think that germline gene editing is worth the risks. This way, some of the benefits can still be achieved while risking a whole lot less. So, in summary, we talked about somatic cell gene therapy versus germline cell gene therapy, and we remember that somatic cell, the changes die off with the individual, and germline, it goes on to your descendants. We talked about some of the advances in recent years and why we really want this technology to work. We talked about what's stopping us from utilizing this technology right away, and that is morality. And then lastly, we talked about different alternatives that you can take to genetic engineering to have a healthy child. So I will leave you with the question that you answered at the beginning. If you could pick the traits of your future child, would you? And I'd like you to answer that again, and possibly raise your hand if you changed your mind throughout this presentation. Thank you so much for listening, and if you have any questions, I will take them now.